an authentic introductory biology experience. So this is so authentic, I haven't looked at this file since I last taught this particular one in 2012.
Wow.
So you know it's the real deal because I didn't even change the date. So that's how you know. So at this point, this is about halfway through the introductory biology sequence. We've spent more time than students ever wanted to on studying the different aspects of biomolecules. So we've talked about what carbohydrates are, and nucleic acids are, and lipids, and I'm missing one, Nicole. Proteins, yes, proteins. How could we forget about our favorite friend, protein? So that's led up to this point where now we're actually going to talk about the basic unit of life. So we've talked about these different parts for weeks on end, and now we're getting into a section on cells. And this is just one of a series of lectures about cells. So at the beginning of every lecture, we start by talking about what it is we want, and by we I mean TAs. I'm not being odd or that odd. What we want you to know are the following objectives. So at the end of today, I want you to be able to draw a generalized prokaryotic cell. So to be able to do that, you'd need to know what the heck a prokaryote is and something about that cell to be able to draw one. Then I want you to be able to compare two different kinds of cells. So these are prokaryotic cells, this first type, and eukaryotic cells, a second cell type. And then I want you to be able to draw and describe some major organelles found within a eukaryotic cell. So these objectives are what you should look at when the night before the exam you feel like sending me an email saying, what is it I need to know about that? This is what I want you to know about that. So first off, just grab a piece of paper. And the first thing I want you guys to do is to draw a cell. So whatever comes to mind, whatever you think a cell is, even if you think it's a prison cell, just draw whatever you know about a cell. Bear in mind, at this point in the real class, you've already been around a while, so you know it's biology, and we will have talked about a whole bunch of different molecules in a cell. So my hint for the actual class was, well, where did you find all that stuff, like the lipids and the proteins? What kind of processes have we already talked about? So for now, just start off by drawing a cell and labeling your cell.
Cell body.
Yeah. Mm-hmm. So anything you can think of, add it in. So for anybody who's done, go ahead and try to draw some different kinds of cells. We're going to go ahead and talk about some different types of cells. What kind of cell is it that you drew? What could other different kinds of cells be? As examples, there are bacterial cells. We also have plant and animal cells. So if you've drawn one or one of these kinds, what was it? And can you draw another kind? And do feel free to share with your neighbor, compare what they have, see if you've got that's similar or something different in those pictures. actually drew phospholipid bilayer. Well done. Yeah, you don't give yourself a lot of room.
That's why we're not.
I couldn't even find it.
See, I even remember.
It's been 4 years. I've been wrong for this.
Cool, where's the lines? Nice. Yeah, exactly. That's right. Yeah, you got it. Cool.
You guys, is it for cellular structure, the skeletal structure, is it microtissue, intermediate tubules? The phospholipid bilayer is the cellular structure, and then transport proteins are like
Okay, so let's see. In terms of types of cells, let's make a multiple choice. Let's pretend we've set up. Have you guys talked about what clickers are?
Yeah.
I don't see you have those, but you've talked about what they are, so you'll know what we're
I
getting
use
at.
clickers in chemistry. They have it in the screen.
Okay, so here's some different cell types, and that's always the answer for people who aren't listening. So what I want you to do, you just put your hands up for the different letters. What we do in class is everybody has a clicker, and you can just respond on your clicker like a remote, and you'd respond if you had drawn a plant cell, you'd respond A, and then we get this little box on the slide that would highlight how many people answered A, right? So for now, how many people drew a plant cell? Right. How many people drew an animal cell? How many people drew bacteria? Just one? You half drew bacteria? Okay. Anybody in there not know what we're doing? Right? Okay, cool. There's always one in every class. All right, so what we're going to do now is we're going to go forward and talk about two broad classes of cell types. So the first way we can classify types of cells is just based on shape or morphology. The biologist way of saying shape. Now there are, in these two groupings, the first grouping, those are prokaryotes. So prokaryotes, an example of that are the bacteria that none of you drew. They're very underrepresented in this group. If you drew one of these, hopefully you drew the absence of a big circle in the middle of it. That circle is the nucleus. So prokaryotes don't have this thing. An example of eukaryotes are us humans. We are eukaryotic, and all of our cells have a structure called a nucleus, and we're going to talk about that a little later. So in these two groupings, the prokaryotes have no nucleus, the eukaryotes do have a nucleus. And at this point, I'd be referring you back to all those chromosomes we spent about a week and a half talking about when we talked about nucleic acids. That's where you find them in the nucleus in eukaryotic cells, like in our cells. Now, using some really fancy technology, using sequence data, where you compare the DNA sequence from organism to organism. Again, we've talked about the structure of nucleic acids in a lot of detail already. You can further classify life into three domains. So the prokaryotic domains are bacteria and archaea, and the eukaryotes, like us, these are eukarya. So these are three domains that get referred to in our reference textbook quite a lot. 2 of those domains are prokaryotic. Those are the guys that have no nucleus. So a little bit about these prokaryotes. The prokaryotes inhabit the widest range of environmental extremes, and one of the unifying rules about prokaryotic cells is they're a lot smaller than our cells. And if you had the pleasure of taking this course with me, then a couple days from now we talk all about microscopy and scale. So instead of just memorizing prokaryotes are real small, well, we'd be looking at what that really means when we compare cellular scale. But alas, you'll be on the beach somewhere. Too bad. Okay. Now, one of the other aspects of prokaryotes is the fact that they are single cell. So sometimes you might find these things growing together in clusters, but they're not multicellular like us. Each one of these cells can survive on its own. So prokaryotes are unicellular. So here's some of the common features of prokaryotic cells. So if on an upcoming exam you were asked to draw a prokaryotic cell, that's your hint. This is one of those things I'd like you to know. All prokaryotes have a plasma membrane. So if we take a close look, this is an electron micrograph. So this was generated by first cutting a cell really thin, and then we use electrons to throw at our sample, and then we collect the electrons that get scattered off of that sample. Any of these regions that are really dark in color, those are said to be electron dense. That's where a lot of electrons hit and bounce back. So this dark line all the way around the cell, that's the plasma membrane. All prokaryotes are going to have a plasma membrane. The plasma membrane is made up of phospholipids, which a lot of you were drawing on your diagram of a cell. These prokaryotes, they don't have a nucleus with all the chromosomes inside, but the DNA is found in a specialized region that we call the nucleoid. And finally, all the prokaryotes have a cytoplasm. So the cytoplasm is a fluid inside the cell, and that's where molecules that we've talked about before, like when we talked about how we make the proteins with ribosomes, we find ribosomes in the cytoplasm. So for now, are there any questions either about the content that at this point you're not so worried about, or this kind of class? Yeah?
Do you post your PowerPoints to Blackboard?
Not only do I post the PowerPoints in Blackboard, but I'll post the whole preceding course in Blackboard. So the slides and the MP3s of the lectures and things like that from the previous term will already be there. Any other questions? Yeah?
The liquid portion of the cytoplasm, does that just like consist of water or?
Oh, that's a great question. In part, it's composed of water, but everything that's dissolved in that water as well. So ions, the ions that we don't really talk so much about in intro bio, but if you go on and take physiology, these ions are really important for maintaining the ability of a cell membrane to conduct electrical information in particular cells. All those ions are dissolved in fluid. All these different proteins that we would have talked about, like enzymes that catalyze reactions, they're dissolved in the fluid. So there is water there, and it's more than just water. There's a lot of stuff dissolved in it. Any other questions about anything? Do you have a question?
Yes, do you prefer students like read over the material under discussion before or after you go over it in class? Like, do you want them to come in prepared, or do you want like your presentation to be the first exposure to it and then the overview?
I prefer that students be prepared. I also prefer a raise, and the odds of the raise are really low. So I'm realistic about how prepared students are when they do come in, which means some will have read ahead, but most with good intentions have either partially or not really spent a lot of time with the reading. And I know that from anonymous surveys of the class and that sort of thing. Yeah?
Are laptops allowed in class?
Yeah,
What about for
not
you?
all classes though. So yeah, in mine, and the TAs go around with a big stick and poke you if you're on Facebook or whatever, but.
What about food in the class? Can you eat like a little snack?
So some classrooms, depending on the location on campus, will have a sign that says no food allowed. And sometimes it gets followed and sometimes it doesn't, from what I can see. When I teach physiology at the very end of the day and people clear out and there's nobody coming back, I'm routinely picking up all sorts of trash of the day. So people are bringing in food and leaving it. Coffee cups, and they didn't bring me one. They leave half-empty ones. It's very sad. Yes?
How much writing goes on in biology class?
How much writing?
Yeah.
With respect to what? Note-taking, answering questions?
For grades, like.
The classes have gotten quite big. So now the intro bio classes are about 100 students. So typically there might be activities that would require, say, a short answer. None of the instructors are asking students to write anything in the form of an essay. So I wouldn't say the course is writing intensive and it's not categorized as one. Mostly because there's just too many people.
So the tests are mostly multiple choice?
Yes, so depending on the instructor you have, there might or might not be some form of short answer, but no matter who you take it from, the short answer would be a fraction in the total exam. Okay, so one of the cool things that these prokaryotes have is an extracellular part of DNA called the plasmid. And the reason why the plasmid is cool is because it's not part of the main chromosome. And it can carry different kinds of genes, including antibiotic-resistant genes. So Biology 142, we go into a fair amount of detail. Wasn't that fun, Nicole? Okay. About how bacteria do it, and they can transfer these plasmids from one cell to another directly. So when bacteria have plasmids with antibiotic-resistant genes, they can exchange these plasmids, so the population of antibiotic-resistant bacteria will grow over a very fast amount of time. And clearly any of us who've ever gone to a hospital or might want to work in one are quite interested in how antibiotic-resistant bacteria arise. Nicole doesn't care, she's doing her homework. Okay. Another cool thing that bacteria can do, and only some eukaryotes can do, well, only some bacteria can do this, they can photosynthesize. So bacteria don't have membrane-enclosed organelles, they don't have chloroplasts. Those are the membrane-enclosed structures that plants have containing all of their photosynthetic pigments. But some bacteria have this folding in of membranes that contain chlorophyll pigment needed to photosynthesize. So isn't that neat? Bacteria can photosynthesize. So another structure that nobody drew here, but usually when I do this, somebody has drawn it, even if they've drawn clearly an animal or a plant cell, are big long flagella. So flagella are prokaryotic structures. They're said to be tail-like, and their main function is for moving around the cell. So we do have some structures called cilia. Cilia are found in cells that are specialized for secretion and absorption. So for example, in our gut is highly specialized for absorption. So they have these structures in their membrane that stick out, and that just increases surface area for absorption. But cilia are different, they have a different function, and they're not moving our gut cells around, right? They're just to make the membrane bigger. Whereas flagella are designed, did I say that? No, they're here in order to move the cell around. Bad biologist, it must be the middle of the summer.
We won't tell anybody.
Thank goodness. Now, another structure you can actually see in this electron micrograph is this really thick stuff outside the plasma membrane, right? That's a cell wall. Our cells don't have cell walls. Plant cells do have cell walls, though different structure. A lot of prokaryotes have cell walls. And there's a really good interest, it's the end of the day, it's 3. A really good reason for us to be interested in the cell wall is the cell wall is a structural component of a bacteria. And remember, bacteria can be pathogenic. So if we want to target these bacteria and break them up, because we don't want bad bacteria growing inside us, we can use an antibiotic, and a lot of antibiotics function by targeting a component of the cell wall. So if you break up that cell wall, then you can kill the bacteria. Isn't that cool? So any questions about anything so far? More questions you ask, the less I talk, it's all good. And as you see, it's going downhill over time. So, yeah.
Animal cells, like humans, our cells don't have cell walls. And like a cell wall, like, is it just more protective? Like, it's kind of like more protective. And I was thinking that if we had cell walls, then like maybe fewer things get into the cell. And like, because like viruses always get into the cell, like maybe that extra layer of protection could like make it smaller.
Yes, that's a good point. And plant cells have cell walls, but one of the difficulties with having a cell wall is being able to move the cell. And okay, our skin cells up here, they're not really moving around amongst themselves, but we do have classes of cells that require motility, and there's a trade-off between motility and having a cell wall. So, yeah. So the prokaryotes have these big flagella to move them around, and that helps them move around. But yeah, I think the reason why we don't have cell walls is really motility. So, should I get into this? I'm not sure. You might like this. Okay, so in the nervous system, there are 2 general classes of cells. There are neurons and glia. You've probably only heard of neurons, right?
Yeah.
Anybody heard of a glia before? Really? 4 people have heard of glial cells? What do they do?
Oh, I don't know if they do it.
Okay, yeah, what do they do?
Aren't they just support cells? They provide like nutrition to the neurons or structural support.
Insulation, okay.
Yeah, I think it's insulation.
Oh yeah, that's right, Cons.
Myelin sheath.
Okay, so there are in the central nervous system 4 kinds of glial cells. One kind is a horrible name, it's called an oligodendrocyte. But that is a myelinating glial cell. What it does is it insulates a neuronal cell. And that's really important because a neuronal cell conducts information in electrical form to a particular target. And if you insulate it, that information will travel quickly. And that's kind of important if you think about information moving around in our brain, right? You want it to go quickly. So at any rate, when we think about the developing nervous system, here's my neuronal cell. So here's its nucleus. That's the cell body, so there's cytoplasm in there. Yes, I can spell. Okay, good. That's the nucleus. So far, does this look kind of like some of your cells? I mean, you could draw better, but you probably drew a circle or something like that. Okay, now, here's something that I don't think any of you drew.
Yep. Axon.
Yeah. So did any of you draw an axon? You drew an axon? Oh, I wish I saw that. Okay, so this is an axon. That makes me excited. Okay, so when this neuron sends information, it sends information along this very long structure called an axon. And how long can these things be? I'm a bad example because I'm short, but the cell body of the neuron is in the central nervous system. It's in the spinal cord. So those little muscles in my toes that if I were really lazy and wearing flip-flops, you'd see me wiggling them. To get those to move, there's a signal being sent from my spinal cord all the way down and the axon to innervate the muscles in my toes. Now, isn't that cool? Now imagine if I were like 2 feet taller and on a basketball team. I'd make a lot more money and I wouldn't be doing it, but my axon would be that much longer, right? So, here's a question for you. Early on, when I was just a little embryo developing, how did the axon know where to go and make a connection with the target? How did it know to innervate a muscle somewhere down there? All right, one thing that happens are these glial cells, they secrete chemicals, and they can secrete chemicals called, you'll love this, chemoattractants.
Sounds sexy.
All right, so that's an attractive chemical. See, it makes sense. And it tells the axon, hey, come on over here. And there are other cells in the environment that are chemorepellents. I'll just abbreviate. And those tell the axon, no, don't go over here. And if the axon doesn't, it just collapses. So these are 2 cells in the nervous system. And we got onto this so long ago, I don't even remember where we started. Drew, where did we start?
You were talking about different cell types, and you were talking about support cells. And you mentioned one, and then it kind of stopped.
Yeah. Oh well. But wasn't that a fun story? It's summer, so we can talk about any biology stuff we want. Now, this is actually a little more human physiology than intro bio, but I thought you'd enjoy that. I always do. All right. Now, one other thing that we find in these prokaryotic cells is something called a glycolipid layer that's found outside the cell wall. So again, this allows bacteria to do things like stick to a substrate, and that can be bad if that substrate is in us and it's in a place we don't like. So we can target the makeup of this capsule to try to eliminate the prokaryotes. This is just here to show you that outside that cell wall, yeah, there actually is stuff. So there's some graduate student out there who spent hours and hours and days cutting very thin sections through material that's so small you can't actually see where the cells are. And then they go to the electron microscope and look, and most of the time they don't get anything. Can you imagine how happy that person was when they finally actually cut right through the middle bacterial cell?
I don't think they can.
I don't think you can because you're laughing at the grad student, but that's how hard this is. That's why I'm sure this picture that you just ignore in the book took some grad student like 5 years to get. And I'm just going to go like this. And you want to look at this cartoon that somebody did in Photoshop in about an hour. Okay, well that cool picture is just showing you all these different layers outside a prokaryotic cell. And that's important because, well, we don't have capsules and we don't have cell walls. So if you want to design a drug that's going to target bacteria colonizing the back of your throat and might kill you, then these are potential targets for drugs. So that's why being able to compare these cells is really important. Any questions so far? We can talk more about the brain again.
Yes.
I like not having an agenda to adhere to. We teach like this all the time.
It is nice.
Okay, so far we talked about prokaryotes. They don't have a nucleus. They do have some funky structures like a cell wall. They have that slimy mixture outside that cell wall. They're a lot smaller than these cells. Eukaryotes range from algae to giant trees, right? So we have a fair amount of diversity when we think about eukaryotes. Most eukaryotes, like us, are multicellular. So we're composed of lots of different cell types. There are unicellular eukaryotes, so this is not a main difference. Anybody know a unicellular eukaryote? Yeah.
A protist?
A protist? Protist. True, protist.
I don't think so.
No, the protists are prokaryotic. Yeah. There is one I'm sure you're familiar with. Anybody like to eat bread?
Yeast.
Yes, so yeast, the yeast we use to bake bread, drink beer, brew beer. Again, how many of you are 21? I'm Canadian, so we can drink at 18. Maybe I'm advertising for a Canadian school now. Well, you guys are already accepted here, right?
We're all
Yeah, so you're trapped. Okay, good.
freshmen.
So at any rate, yeast that we take advantage of, yeast, when it undergoes fermentation, a process we would talk about in Biology 141, that fermentation process is vital to being able to bake bread and to brew beer. And that's an example of a unicellular eukaryote. One of the major challenges of this hard eukaryotic life we all have is the fact that the large size of this cell makes it hard for stuff to diffuse in, like oxygen. You need oxygen to get inside these cells. And one of the things that eukaryotic cells do in order to meet this challenge is to have some organization in the cell. So eukaryotes have structures called organelles. Organelles compartmentalize, I said that right, particular enzymes and such, all related together for particular processes. So I mentioned a relationship between oxygen in the mitochondria. Inside these mitochondria, how many people have heard this horrible term of they are the work or powerhouse of the cell? Yeah, isn't that awful? Anyway, there are enzymes required for aerobic metabolism inside the mitochondria. All those enzymes are contained inside this membrane enclosed structure, and in 141 we would talk about that process in the mitochondria. Any questions about anything so far? Okay, let's vote again. How many people feel like this is what they think a biology class would be like, and B is no? How many people vote yeah? Is this what you expected a biology class to be like? You didn't expect it to be this much fun? Okay, how many people feel this isn't quite what they expected for biology? Okay, so what did you expect?
My freshman bio class was extremely boring.
Oh, okay.
And I don't know, I was just never a fan of biology, but I loved anatomy senior year. So, I don't know, I'm liking this
Okay.
a lot.
Cool. Do you have your hand up? Oh, okay. How about you? Did you? I think there's somebody else over here that had their hand up or no. Anybody else? Expectations? Met? Not met? Yeah.
I put my hand up for yes, but my AP bio class had a lot of math in it, and I was wondering how much math is in the introductory bio class.
In intro bio, in the lab, there's some more math than the lecture. There's a bit of math in physiology, but not really a lot of math in intro, and that's just because of topic, not because we tried to get rid of all the math or anything. We just don't address a lot of topics that are mathematical in intro bio, but you would use math a fair bit in the lab to do different calculations, definitely. Yeah, yeah.
So this is like the first lecture you actually did?
Oh no, this is lecture 21 out of about 40. Yeah, so we are stepping in midway into the course. This is what got presented in lecture 21. It did not change a thing. That just tells you I didn't have any time on the weekend to change it. Yeah. So this isn't day one, so if it's a little overwhelming, this isn't the very first class.
No, I actually thought it was going to be a lot harder than this. I never thought you would go through the actual self parts. Like to me, that's like basic knowledge.
So here's the thing. One of the things that we deal with in intro science classes are the different range of preparedness of students and the misconceptions that students bring in. And the purpose of the intro classes is to try to get everybody on an equal playing field. And what you find is in 141, it is pretty general and basic. 142 is quite different from 141. It goes into topics that you would never see in high school, like I guess we were just talking about one, and that was how different bacteria can mate. Anybody heard of conjugation and transduction before? 1, 2, 3. Oh, good. Excellent. Then you'll ace 142. Population genetics.
Yes.
Excellent. Now, in those topics, we do tend to go into a little further detail than what people have heard. So, you could probably tell me what the Hardy-Weinberg formula is. I bet you I could find some questions you couldn't answer though. Yeah. So one of the feedback we get says, yeah, 142 was no problem. I did AP Bio and aced it, whatever. And 141 is pretty similar. And then people tell us 142 is quite a bit more difficult. A lot of topics they haven't necessarily heard of, or if they have, they didn't really get it. So it gets more challenging pretty quick, but it's not so hard off the bat. Yeah.
So, in your opinion, what's the final aim on what kind of 141 like getting yourself prepared for 142? I guess at the end of lecture 40 with like the final, like, is this just the depth of what you would need to know to do well in 142?
It's a challenging question for me to answer, so I might sound a little rambly because. The content in 141 does not feed directly into 142, but people who AP out of 141, so they never take it, tend to have a more challenging time with 142, and it's probably for a large number of reasons. So one is you don't know any of the bio instructors, you don't know who to go with, you haven't taken the bio lab before, so you're just not into that rhythm. Part of it probably is content. So if you took bio 3 years before, it's not fresh in your mind. So people do better if they go through the sequence, but I don't necessarily think it's 100% the content. I think there's a variety of factors contributing to that. Content is just one of them. Yeah?
What is the pace of your bio class? Like how many chapters would you cover in a certain amount of time before you took an exam?
We generally have a weekly assignment that's done online. We have a midterm about a month of the way in, another month after that, and then one more month and it's a final. So how many chapters would be on an exam? Probably somewhere around up to 5 sections. So in this section, this is prokaryotic, eukaryotic cells. It'll take maybe 3 lectures to go through. That might be one segment, and there'd be 4 other different segments. So I think it's a reasonable amount. Lots of people do well on the exams, but not everybody. And not everybody who doesn't do well knows that they were going to be the one who doesn't do well, which is one of our challenges in intro courses in particular.
And bio lab doesn't follow bio lecture, right? Like, are they just separate?
So, not only do they not completely match, but I'm not sure if it's this fall or the next fall, but they'll just be completely separate credits. So, yeah. Yeah. Any other questions? Yes?
What's your favorite topic to teach?
Nervous system physiology
Yes.
and human physiology. That's probably my favorite topic to teach. Oh, and today we did lecture on muscle metabolism and fatigue. So we got to look at athletes crossing the finish line in the Hawaii Ironman and dropping to the ground before they could make it. So yeah, that's a pretty fun one too. So you hang on through intro and then you get to take physiology. That's really cool. Yeah. Any other questions? Okay, so we can do some more bio. Here's some key differences between eukaryotic and prokaryotic cells. Now, something to keep in mind is, remember at the very start when I told you the objectives, and I said I want you to be able to draw a prokaryotic cell, and then I told you about stuff in the prokaryotic cell. The purpose being I would like you to be able to put it all together and draw one. And if you could do that, you could answer any question I would ask you at the exam. The second objective was I wanted you to be able to compare prokaryotic and eukaryotic cells, and hey, here we are, right? So this is how you know what to study. Look at those learning objectives. Almost every instructor I know at Emory uses learning objectives in one form or another and gives them to you. So it should help direct your attention as you're studying. So this is a table straight out of the book that we use, and these are some highlighted features distinguishing prokaryotic and eukaryotic cells. We've already talked about some of these. So the eukaryotic chromosomes are found in a membrane-enclosed nucleus. The eukaryotic cells are much bigger. Eukaryotic cells have a whole lot of internal membrane, and we're going to talk about that maybe today, or maybe when you take Biology 141 sometime. And eukaryotic cells have a diverse and dynamic cytoskeleton. So in order for a cell to have a big, long, specialized structure like an axon, this is the axon. Information in the form of an action potential gets conducted that way. This is my little diagram for an electrical charge. There are collections of proteins underneath the plasma membrane of this thing to give it shape. In fact, this glial cell was secreting chemicals called chemoattractants that bind to receptors on the end of this axon. Their whole purpose is to modify the cytoskeleton. That's how this axon grows towards a target. It's all about regulating the cytoskeleton. So eukaryotic cells have extensive amounts of cytoskeleton that can be regulated to make these kinds of structures like the neurons in our nervous system. This is just a cartoon of an animal cell. This is like what a lot of you drew. A lot of you drew some of these structures, like this is endoplasmic reticulum. The rough form of endoplasmic reticulum is called rough because it's got all these ribosomes that are drawn in little red, orange dots here. There's also smooth endoplasmic reticulum that's not specialized for translation of proteins. So if we were going through this section, these are structures that we would go into a little more detail on as we go forward. For now, I just want you to compare this general animal cell to a general plant cell. This plant cell, like some of the prokaryotic cells, is a cell wall that's drawn here in yellow. Plant cells have these structures called chloroplasts. The chloroplasts are membrane-enclosed organelles. The membranes inside the chloroplast contain the enzymes needed for photosynthesis. Also, we find in this structure a vacuole. This is a big fluid-filled space that a lot of waste gets transported into. And as a general comparison, here they've drawn a bacteria. Comparing that to this plant cell, the bacteria is much smaller. Okay. So in the class we would go on and we talk about some of the different structures, and many of these are structures you would have heard about before. So a lot of this material probably wouldn't be new for you. This is a figure of a nucleus. This is the real thing. That's a cartoon symbolizing the nucleus. Note this double membrane. And another really important structure we don't think about when we think about the nucleus is the fact that the membrane has pores. All of those messenger RNAs that we will have already talked about in this class need to get out of the nucleus somehow, and that happens through specific pores. And this is a structure you'll probably heard of before. This is the real thing. On our electron micrograph in black, this is membrane, right? And also on this electron micrograph, you see these dots. Those are ribosomes. The ribosomes are translating new proteins. Also in this figure, they've shown us some dots down here, right? There's a whole bunch of dots down there. Those are free ribosomes in the cytoplasm. So I've got a question for you, but I'll answer yours first.
Why did you decide to teach instead of do research?
There are some real long reasons for that are better suited for the pub, but I just always liked teaching more. When I was in high school and in university, I taught swimming and first aid and CPR and loved it. I liked research, but I didn't love it as much. For a real, more complicated answer, you could look at the job market. So my question for you guys is, picture the ribosomes that are found in the rough endoplasmic reticulum, and then picture those ribosomes that are in the cytoplasm. Are they the same ones? Is it a distinct ribosome that's found down here? Or is it, well, just the same kind of thing that you can find up here? So what do you think? Are they the same or are they different? So how many people vote for they're the same? 1, 2, 3, 4, 5. How many people vote for they're different? Okay. Now, good for you voted number 8A. Yeah, they are the same thing. And guess what? They can even interchange. Right? So the ribosomes you find down here in the cytoplasm, they're the exact same thing as the ribosomes we find up here attached to the ER. Take a minute and think about, well, how did some end up down here and how did some end up there? And if you have no idea, ask your neighbor. Maybe they know. What do you think? Okay. What's different between them? What's that? Yeah, okay. What else is different about them? You're right. That's still okay. Okay, what's the difference between knowing that?
Yeah, that's what I'm thinking.
No, they only Are the proteins all the same? Or are they well, there's something that the protein source translated that dictates where the ribosomes Okay, so first off, why are some ribosomes found in the cytoplasm? What do you think they're doing? All right, they're translating proteins, and what are the ribosomes up here doing? Right, they're also translating proteins. So this is what they have in common. They're all making proteins. Are they all making the same proteins? Okay. Again, yes. They're all making the same kind of proteins. B, no. What do you think? How many people vote yes, they're all making the same protein? 1, no idea. 2, 3. Vote with confidence even if you're not. firmly believe what you're not sure about. I'm not American, so I'm just getting used to politics down here. Okay, B, what do you think? B? No, I'm not saying. Okay, so if you voted B, no, they're not the same proteins. So all these ribosomes are making proteins, but they're not making the same kind of protein. They're making different proteins. Proteins that are going to function in the cytoplasm are being made by ribosomes in the cytoplasm. So if you're a cytoplasmic enzyme, for example, and you're going to do your work in the cytoplasm, then the ribosome, when it translates you, it's going to go down here in the cytoplasm. Particular kinds of proteins end up being made in the rough endoplasmic reticulum. Those include proteins that will be secreted from the cell. So what gets secreted from the cell? Well, one of the major communication systems in our body is the endocrine system. Right? Sound familiar, folks? Okay. Does the pancreas sound familiar? Okay, yes, so far so good. Okay, so there are specialized cells in the pancreas called beta cells in response to having high blood glucose levels. Then these beta cells are going to secrete a protein hormone called insulin into our blood. Right? So insulin is a protein. It gets translated inside the beta cells in their rough endoplasmic reticulum. So when a ribosome gets an mRNA and that mRNA encodes for insulin, this ribosome knows to go to the ER because proteins that get secreted from the cell get manufactured in the rough endoplasmic reticulum. So where a ribosome goes really depends on what protein it happens to be translating, and that's how you get some ribosomes in the cytoplasm and some ribosomes in the rough ER. Any questions about any of that? Yeah?
When you said that they're interchangeable, do you mean that one ribosome can manufacture like several different proteins?
Yes, not at the same time. But at one point in time, the ribosome could make a cytoplasmic protein, and then the next protein it translates could be a secretory protein. Yeah. Yeah.
So if they can create some proteins, then wouldn't damage it? It's just depending on the specificity if it's in the cytoplasm or in the ER, right?
Yeah, so the ribosomes are the same. Yeah, that's right. Yeah. Any other questions? Yes.
How are they like given, like when they're making a different protein, how do they get their assignment?
Oh, that's a great question. So when you are making a protein and you are joining together amino acids in a string, basically, and we would have already talked about the details of this, not all those amino acids are going to make it into the final protein product. So imagine like when you make cookies and you're going to cut the cookies out with cookie cutters, you're starting off with just this sheet of cookie dough. It's not your final product. To make your final product, you've got to cut off what you want, and then you get this excess left behind. Proteins are the same. You can make immature forms of proteins that you then cut parts off of. So using insulin as an example, insulin gets made in this immature form that we then cut some parts off of. One of the parts that we cut off is a signal sequence. So when the ribosome is translating this new protein and it synthesizes the signal sequence targets the ribosome to the rough ER. So without the signal sequence, then the ribosome can't go there. So the signal sequence is a series of amino acids that says, hey, take me to the ER, and it does. So one of the cool things you can do to test this is you can take those amino acids from a signal sequence, attach it to a protein that would never go to the ER, and once that protein has a signal sequence, it will go to the ER. So that's how we know the signal sequence does this. Yeah? Yeah.
Okay, pardon if this sounds like, oh my god, did you seriously just ask this question because my biology is so absolutely terrible?
Cool.
But like, you know how oxygenated blood travels through the body and then, you know, it gets it travels to each independent cell. How does the cell extract oxygen from the bloodstream?
That's a great question, and it depends on the cell type. So you said, how does the cell extract oxygen? Some cells can do that. So for example, skeletal muscle has a protein called myoglobin, and myoglobin is very high affinity for oxygen. Myoglobin, under physiological conditions, has higher affinity for oxygen than hemoglobin, and most of our oxygen in our circulation is bound to hemoglobin. So when the blood gets to the skeletal muscle, there actually is something with higher affinity for it that will help take it up. But most of our cells, they aren't pulling the oxygen out, they're just using oxygen. And when the blood goes to a region of lower oxygen concentration, oxygen is just following its concentration gradient, going from a region of high oxygen concentration in the blood to a region of lower oxygen concentration in the tissues. So with oxygen following a concentration gradient, it's not really being pulled by anything. Is that the long, complicated way of answering your question?
I'm not really sure.
Okay, cool. Any other questions? Yes?
Can I ask a general question
Why
about
not?
the glial cells?
Yeah.
So do these cells die after development is over? Like why would those cells turn back on if somebody says rips a nerve?
That's a fantastic question that we could go to a conference about.
Sorry.
No. So and there were a number of questions. So one of the questions, do the glia just die after development? The answer is no. Aiding in development is just one process that glial cells accomplish. We talked a little bit about this insulation of axons, and there's a class of glia that will cover the axon to prevent ions from leaking out of it. So you need them in adulthood, and in diseases like multiple sclerosis, you lose the myelination and then you lose neuronal function. So yeah, you have to have them. What else did you ask? So regeneration is really complicated. One of the bad things that happens to prevent regeneration in our central nervous system is the buildup of scar tissue that you need to remove. Some of those growth factors we talked about are only expressed in early development, and then they're not expressed anymore. So in order to regenerate, you have to tackle like that scarring. You'd have to get cells to start secreting factors they've stopped secreting and that sort of thing. So it's a really complicated question. Yeah. Any other questions? Yeah.
Say that the glia cells never stopped secreting what they were secreting in early development.
Mm-hmm.
What happened theoretically, obviously?
So another great and complicated question. One of the complicated things that happens in biology is signaling molecules tend to be reused by different cells. So one great example is, and you'll love the name, it's a molecule called sonic hedgehog. Right? You've at least heard the name even if you don't know what it does, right? So it turns out this protein is involved in early spinal cord development. It's how particular cells know to become neuronal cells. But it turns out it gets used in other parts of the body too with different functions. So if you've got a glial cell that keeps secreting a factor that normally it stops, well, these factors might be used for other processes, and who knows what's going to happen, right? If you've got cells that are continually expressing these growth factors that normally aren't expressed later, you're probably activating other signaling pathways and causing like chaos. So that's one thing that could happen. Any other questions? has a chance, he can ask me anything, and I'm not getting paid, so, you know, I can tell you what the honest answer, yeah. If Drew goes to the bathroom, I can tell you the real honest answer.
I'll just tell him. And,
Yeah.
Typically, how many people fill the info forms?
No, it's a fair question. Out of 100 people, at least in mine, 1 to 3. So a very small number, but I don't think I've ever had a year where there wasn't somebody. Yeah.
How many people fail being a C or lower?
That's a good question, and it varies. I mean, in my section, what I tend to observe is, so this is number of students,
He's gonna do the dog.
and if this is A, B, C, D, F, the curve is kind of like that.
Oh,
Alright, that's not a normal distribution. A normal distribution would look something like this.
oh, that's a pretty good
Right.
statistic right there. That's cute.
So,
Right.
lots of people get A's and B's. Not that many people get C's and D's. Now, here's the trouble, and this is probably something you've talked about at great length already, and it's the people who end up in this box on day one would never have thought they would have been, and they don't understand why. So, I show you this to say, hey, on my end, you know, things look pretty good. But this is a real hard place to both be in and to work with, and you got to be really careful because nobody down here ever comes in and says, I'm going to get a D in biology. Just the way it goes. So all that said, the way you prevent that is by not just working hard, but working hard the right way. When you get a great grade on midterm 1, not just like not coming to class ever again. Those people don't usually do that well. It does get more difficult as we go forward. And as you can see, like we're starting with some stuff that most of you are pretty familiar with. But it will get more complicated. And we tend to ask questions on the exam that aren't all just things like the form of endoplasmic reticulum that ribosomes bind to is, and then the answer is rough ER, you just memorize that. Right? So we tend to ask more challenging questions than that, see if people really understand it. Yes?
Are bio-exams multiple choice or additional answer?
They tend to be, depending on the section you're in, multiple choice, sometimes with the addition of something else. So maybe with a few questions that are short answer. Often they're just multiple choice, just because it's the only way for one person to give an exam to 100 people and get their grades back in any reasonable amount of time.
Do the other bio professors like recorded their lectures and put them up online like you do? Or?
Not all of them. I know some do, but not everybody does that. Yeah.
AP Print covers just 141, right?
Yeah.
How effective of a supplement is that for the actual 141? You just go straight to 142, is it?
It can vary widely. Typically, people report back that they find 142 a lot more challenging than 141. And some people do well in 142, and then some people who AP don't, and it's a mix. So it's hard for me to say, because AP programs can vary quite a bit, and some people might not remember stuff. Not every AP program is the same, so it's hard for me to generalize. Some people do well with it, and some people don't, I can tell you that much. Yeah.
So last week we watched a film called Outbreak. I don't know if you're familiar or not. It's essentially this infectious disease infects a population in Africa, and then somehow it manages to get to the United States. And at the beginning of the film, a Nobel laureate was quoted. I forget his name, but he said essentially that the virus was the most dangerous thing to mankind. And I was just curious, from a biologist's perspective, would you agree with that?
Well, not having seen the movie, no, I have no comment to make. I don't know. Yeah, I didn't see it.
Would you just say in general, like, what do you think poses the greatest threat?
Oh, ourselves.
Aren't-
Yeah. We're the only, and I'm speaking broadly and on population levels, so don't take this personally, include me as a human too, but we're the only folks stupid enough to put our environment at such risk where it's the only place we have to live, you know? So, yeah, our greatest risk is ourselves, and we're not doing anything about it. So, there you go. Happy day. Any other questions? So in this lecture, the very last thing we would have done were to come back to more questions. And within my class, and I believe other instructors do similar things, Nicole. So like, do you know what I asked? Depending on the class, we have lots of questions in class, and these can be in the form of multiple choice clicker questions, or these might be more discussion questions and written questions, but I think all of us try to stimulate discussion and some form of interactivity depending on the class and the topic. And those questions, think about it this way, if the instructor is going to take the time to put these questions in there and take valuable class time away from me talking at you, it's probably something important, so you want to know how to answer those questions. Sometimes the instructor gets so tired that they even grab these questions, put them back on exams, instead of spending an hour trying to come up with some new way of asking the same thing. They got the inside scoop for that one. So these questions are very important, right? If they're there, they're about a topic that's really important. And you really want to make sure that when the instructor isn't there guiding you through the question, you can answer them yourselves. And that's a big difference between the people who are here and the people who are over here. Any other questions? Yeah.
Earlier you said that every week there's an online assignment.
Mm-hmm.
Like, what is that like?
So what you do is you log into our learning management system. Isn't that a horrible word? Are you guys on Blackboard already? Yeah. So generally most classes have a site. In Biology 141, 142 will have a site. And within that you can log into assignments. And the assignments are 10 multiple choice questions based on the previous week's material. So some people spend a lot of time and take them quite seriously, and other people log in 10 minutes before they're due at the end of the week and randomly guess at answers. It varies. And I can tell who, because I can see who logged in when and how long it took. Yeah.
Would there be a time limit on that?
I think some instructors put a time limit in. I've never felt that mean. Yeah, I've never had a problem of people spending too long on them. It's mostly people who just go in and don't actually do them. You know, they got the answers somebody else, or they are guessing, or whatever. That's more of the issue. So I don't see why I put a time limit on it when the problem is mostly people who could really use the work. They're not doing that well on exams, aren't taking advantage of the chance to learn how to answer the questions by doing the assignments. But some people seem to want to put a time limit on it. I haven't. Any other questions? Yeah.
Are your classes mostly freshmen and mostly sophomores, or a mix between the two, or what is the general distribution?
It's a real good blend of both. I'd say it's mostly sophomores. Yeah, so a lot of people get advised to not take biology and chemistry at the same time, and are generally advised to take chemistry first. Even people who are going to major in biology and really need the prerequisite to take the other classes, so I don't understand that logic, but, so that means a lot of people take bio a little later. They're the sophomores. And on the class sheet, I'll see there are even juniors and seniors that take it too, but it's mostly sophomores. Yeah.
Regarding students taking chemistry, considering you teach bio, like what has been told to me is that chemistry will be great preparation for 141. Would you agree with that?
For bio 141?
Yes.
Only the first week and a half.
Oh. Okay.
Yeah, besides that, no, not really. There is intermingling of both. Some of the chemicals you're studying in chemistry are actually important because they're in living things. And in biology, in order to understand how the different biomolecules work, like how DNA has the structure it has, and that's important to really focus on its function, we need to talk about things that you're already kind of familiar with, like chemical bonds. But we get through aspects of chemistry like bonds and free energy in about a week, and then we move on. So yes, it would help that first couple weeks if you took chemistry. You'd know more about chemistry than some of us would, because we haven't taken chemistry in a while in those first 2 weeks. But we're teaching what you need to know for the biology part, and we get through that pretty quick. Yeah.
So why do people say that it's better to take chemistry first?
One reason why people advise not taking them at the same time anyway is it is a fair workload. I think it's to get it out of a way if you're a bio major, because you're not going to use that, so just get chemistry out of the way and then do bio. That's my feeling of what other people are advising students to do, but I don't really know.
Because I was thinking, because I wanted to do both, but I don't want to do them at the same time. I was like, oh, bio first. Is there anything wrong with that?
Well, on my end, if you took bio first, when you've got bio 141 and 142 under your belt, now you can take all the bio courses. Because bio 141 and 2 are the prerequisite for everything. And if you're here for 4 years, if you take bio in the first year, now you've got 3 years to take everything, if that's what you want to concentrate in, right?
Is that more beneficial for like an MBB major?
For an NBB major, yeah, because 141 and 142 are the prerequisites to all the NBB courses as well.
Oh, there you go.
So,
Thank you.
now, this is my advice, my opinion, might not necessarily fit everybody, but if I had so many credits to take in upper year bio or NBB, and there was a prerequisite for those two, I'd want to do the prerequisites early so I had more opportunity to fit in all the other courses in my concentration. I don't know, do you have an opinion?
I'm going to put you in a cage with Pat, and you two can have opinions.
Again, I said my opinion, it can
Sure,
vary,
sure.
right, but that's my opinion.
Sure.
Actually,
Yeah.
sure about that, I was told to do chem first and then bio and orgo together. Sophomore year, I've been doing orgo later on with physics. Like that's how it kind of worked out. So it really depends on how you feel about that. Because you need chemistry before you can do orgo. So you kind of mix it that way.
Ultimately, no term is going to be completely perfect, and you'll have to take some courses with other courses at some point because you're only here so long. So whether it's freshman physics, which you're doing now, and organic chemistry, or bio and chemistry, or what, there's going to be some term that will be less than comfortable for the average bio or NBB or even chemistry major at some point.